Hello, thank you very much for this opportunity to show you what we do and where are we located. We are the Lipids and Liver Research Group. This is a presentation of some of us. We have here pictures of us in the Day of Women in Science, the Day of the cholangiocarcinoma, and some of our celebrations in PhD thesis, in Christmas, where we are a group that work a lot together because the aims that we have need the cooperation of all of us. We are located at the Faculty of Medicine and Nursing at the University of the Basque Country in Bilbao, which is just by France. And we also have a lab in the new campus, which is just a research campus in a building called Maria Goiri. Besides, we are also part of the Biocruces Vizcaya Health Research Institute and also a part of the Spanish network for study in liver diseases. In these two organizations, it's really important the cooperation between basic and clinical research. So some of our projects are in common with clinical researchers. So our aim is to find new mechanisms involved in liver diseases so that we can target them and recover from different types of liver diseases. Among them, liver cancer and metabolic associated fatty liver disease and other forms of liver damages are our main focus. Inside liver cancer, we are mainly involved in cholangiocarcinoma and hepatocellular carcinoma, although we also have a project in colorectal cancer liver metastases. So our first aim is to know how the energetic metabolism modulates according to the specific pathology. What is the role of the lipid and lipoprotein metabolism and the crosstalk with amino acids, so that we can find targets of treatment. Specifically, we are also interested in cell cycle regulators, such as several transcription factors that we know that not just modulate proliferation, but also. So why are we interested on energetic metabolism? So as you know, cancer cells need to grow and they need different sources of energy. Lipid metabolism here in green is completely or really related to carbohydrate metabolism, but also amino acid metabolism. So lipid metabolism is like in the middle of the energy metabolism. Why lipids? Well, we know that each cellular type has its own lipidoma. There are more than 1,000 different lipids, and each tissue and organelle differ both quantitatively and qualitatively in their lipid composition. This is really important, not just for physiological functions, but in pathological situations such as in cancer and in liver cancer specifically, lipids might change not just in concentration, but also in localization. So we know that there are several processes, these are the most common ones that regulate how the lipid concentration is regulated in liver. So we have really specific techniques that are really useful to know how these different processes changes. These are classical biological techniques based on the usage of radio-labeled substrates, but we also combine them with lipidomics, proteomics, and genomics. So given that our work and the determination of modulation of different metabolic pathways in vivo and in vitro is a really challenge process, we need a really close cooperation between all the members of our group. So whenever we have an experiment, we need to be all together with all the different organelles or organs to do ex vivo or in vivo the metabolic quantifications. We also use models of obesity, dietary-induced obesity, and also genetically induced obesity, given that these metabolic diseases play an important role in those liver cancers. We are really trained on analyzing metabolic fluxes in vivo. So we also give the mice radio-level substrates to do lipid tolerance tests and to follow the dietary lipids into the body to see how the tumors will do their uptake from lipoproteins. Here we have some examples of the results, how we can follow the clearance of dietary lipids through the blood and also the uptake in different tissues. So an example of this work has been the just published, well, last year we published in collaboration with the Biodiagnostia group led by Chus Bañales. We measure how different targeting fatty acid oxidation in cholangiocarcinoma and specific type of cholangiocarcinoma will lead to the reversion or recovering from it. We are also participating in this cholangionet cost action, and apart from cholangiocarcinoma, we also are interested, as I mentioned, in hepatocellular carcinoma. So we have just found how transcription factors related with cell cycle control also controls metabolism in a specific type of hepatocellular carcinoma. So just want to show, well, that this is our group. Thank you to our group, but also to some of our collaborators. Some of them are here, some of them are not, but for us it's really important the networking that we have built here in this specific topic. Thank you very much.